Hi, my name is Jean-Paul Chatterian. I'm a graduate student in the Burton Jones Lab at the University of California, Irvine.
And I'm Sonia Lombroso, a graduate student in the Bennett Lab at the University of Pennsylvania. We're really excited to tell you about our recently published study in the Journal of Experimental Medicine titled Engineering an Inhibitor-Resistant Human CSF1R Receptor Variant for Microglia Replacement.
Microglia are the innate immune cell of the brain and are highly implicated in neurodegenerative disease. As such, microglia have become exciting targets for drug development in recent years, but also offer an exciting possibility for immune cell therapies in the CNS. However, microglia are highly regulated and engraft minimally in an occupied niche. Therefore, we sought to overcome this hurdle by taking advantage of microglial dependency on CSF1 receptor signaling and clinically available CSF1 receptor inhibitors. We hypothesized that engineering a CSF1R variant resistant to CSF1R inhibitors would allow for the depletion of endogenous microglia and ultimately replacement within the CNS.
Our first step in creating a safe, specific way to replace microglia was to analyze the structure of the CSF1 receptor bound to one type of inhibitor called Plexxikon. So we did this and identified regions in the kinase domain that we predicted could prevent or weaken Plex binding. We then introduced and screened these amino acid substitutions and identified one particularly resistant variant, G795A. We moved this variant into murine macrophages, where we performed in vitro assays to show that it conferred inhibitor resistance and did not have appreciable effect on kinase signaling. Next, we moved in vivo, where we found that G795A expression was sufficient to enable substantial brain macrophage engraftment in both neonatal and adult transplants. G795A expressing macrophages were also capable of sticking around in the brain after the cessation of inhibitor treatment, which is important for translational relevance.
We took a slightly different approach and focused on the crystal structure of CSF1R bound to two well-studied CSF1R inhibitors and identified the glycine residue at the 795th position of the receptor as an ideal candidate for CRISPR editing. We generated an isogenic set of iPSCs that were differentiated into microglia for in vitro and in vivo validation, and likewise found that G795A confers broad resistance to multiple inhibitors. Furthermore, G795A microglia engraft and respond to inflammatory stimuli as wild-type microglia do, and not only proliferate under CSF1 inhibition, but also persist after cessation throughout the murine brain.
We hope our findings here offer a new path for efficient microglia replacement without the need for traditional chemotherapy or genetic modification. There are a number of future studies that can be done to further validate these findings, including more substantial kinase signaling experiments. That being said, we're thrilled by the potential applications for this tool, which include things like correcting mutations driving microgliopathies or delivery of genes to compensate in other diseases, for example, the delivery of lysosomal hydrolases to compensate in pediatric leukodystrophies. Moreover, we hope this approach will broadly enhance the study of human microglia in general. Thanks for listening, and we hope you enjoy the paper.